Okay, all right, it's time. Not many people today. Because it's sunny. Nice day. Wednesday, low attendance. So just taking a break in the middle. Okay, so I put the slide up. So I hope you got some time to get, reviewed. So this is a leaf response, photosynthetic response, if you remember, to the light intensity. And what is the unit of light intensity we want to use for plants? Remember? Anyone? Light intensity unit you want to use for plants? Micromoles per square meter per second. And what do you call that? Photon flux. But specifically what you call that? Photon flux applies to any wave bands, but specific wavelengths starting from 400 to 700 nanometers. How do you call that? Why? You are talking about plants, not human being. Photosynthetic photon flux. Okay, so the radiation is called photosynthetic or photosynthetically active radiation that is driving photosynthesis, but the measurement or unit used for quantifying PAR, photosynthetically active radiation, PAR, is photosynthetic photon flux. Okay, and then unit is micromoles per square meter per second. Okay, all right. So that's the unit you should use in the light intensity. And then the leaf response, single leaf response is quantified by multiple parameters. One is the light compensation point, which is the light intensity balancing photosynthesis and respiration. The second one is the initial slope, which is the slope when light is the only limiting factor. So the maximum response, maximum efficiency in that range. So that is called quantum efficiency. And then saturation point, light saturation point, that is maximizing photosynthesis. Okay? And then that level of photosynthesis is called saturation or maximum net photosynthetic rate. Okay? All right, so what I'm going to do, since it's nice day and Monday, if you remember, it's an overcasting day, so we're going to go to greenhouse now with my sensor, quantum sensor or PPF sensor with me, and then we're going to measure the light intensity. All right, so we go out to the greenhouse. Okay, so of course it's getting cloudy, huh? Of course. So would you like to be a volunteer cameraman? So relatively close to me so that it captures sound as well. Okay, so again, we are outside and I have a PPF sensor with me. So today, right now, it's cloudy a little bit or hazy. And then it's 500 micromoles. Okay, 400, 370, 370, 390. So that's the light intensity, horizontal, almost horizontal. Okay, okay. If I tilt the sensor toward the sun, of course it reads a little bit higher, right? It's 750. Okay, right now. Makes sense, right? Okay, let's go into the greenhouse. It's again 400 micromoles. right here, very south end of the greenhouse. So again, the same position, I'm putting sensor in, and the light intensity here is, oops, it's 158 micromoles. Okay, so that because of the structure, the shade creating, right? So that's why it's a lot less than outside. 150, 143. Okay, so I'm going to measure the inside. You see the row. Cameraman, please, camera person, please go into the row. Yeah, so we are gonna go into the row. All right, turn around. Turn around. Thank you. All right, so I'm gonna measure right here. Okay, so right now I have 10.3 micromoles. 10.3. Okay, huge reduction. All right, so the top of the canopy is probably similar to what I was measuring, or maybe a little bit better, right? But the lower the canopy, underneath, I got less than, right now it's 16. All right, so it's a huge difference. You wanna see the west side of the greenhouse and east side of the greenhouse to get a little bit more perspective. Okay, so I'm moving to west side of the greenhouse. Okay, so I'm standing west side of the greenhouse. West side of the greenhouse we have young cucumber seedlings. Just looks like just planted. Anyone know? Does anyone know when those plants are put in? Courtney, do you know? Hello? Oh, she's somewhere. But anyway, so the light intensity here is 350 micromoles. All right, everyone. 350 in the west side. So it's depending on the location, relative to the angle of the sun, location of the sun, the light intensity is different. And then of course, canopy affecting light intensity. Okay? All right. So we are going back to the classroom. Okay, welcome back to the classroom. Okay, just wanted to check everything is working fine. Okay, all right. Yeah, so you got some sense, right? Greenhouse environment. The greenhouse you were, a few minutes ago is a hydroponic tomato greenhouse, and typical production style we call high wire production. So the tall canopy, so many layers of leaves. Okay. Approximately 3 to 4 plants growing in each square meter. So that's the density, approximately. That's a typical one. All right, so, okay. And the light intensity, so this is a leaf response of tomato, photosynthesis. So now it's ppf, micromole per square meter per second. And then what we are seeing outside was 500 micromoles range, right? 400 to 500. And then in the greenhouse, it was 300 micromoles because it's 4:30, 4 p.m. It's toward the end of the day, of the winter day. And then under the canopy, it's only 10 micromoles, at most 20. So it's very different light intensity. Of course, we have seen east and west difference too. Okay, so here is remember the exercise I said, sort of not a homework, but do this exercise in your head. Think about what and why the difference like this. When you show leaf response and then the canopy response. Okay, before going deeper, let me explain what it is. So the leaf response is the same one as you see, as you saw. X-axis is the light intensity, y-axis is the photosynthetic rate per leaf area base. okay, per leaf area base. So the light intensity is actually hitting that particular light used for measurement. Canopy photosynthesis, light intensity is the intensity over the canopy, okay, top of the canopy. And photosynthetic rate is expressed as, in this case, milligram, which is mass of CO2. CO2 absorbed, and then that amount is expressed in grams, milligrams, per square meter of growing area instead of leaf, because so many leaves, so you can't really say per square meter of which leaf. So usually canopy photosynthesis is expressed per square meter of growing area. So just a section of big canopy. So if you multiply by what? 300 square meter of that greenhouse, then that's the whole greenhouse photosynthetic rate. Are you with me? Okay. And then the time dimension is the same, per second. So the both are basically per second rate, but this one is, canopy one is a per square meter base, so the magnitude of photosynthesis is much bigger. The leaf response is per leaf area base, so it's only one single leaf, and then the 1 square meter size of leaf, you sort of equate that. So that the magnitude is lower, therefore the unit is micromole level instead of milligram or millimole level. Okay, so the magnitude is different. But most important point I want to discuss today, using a few minutes now, is why leaf response is just like our textbook photosynthetic response curve, saturating at certain light intensity. In this case, it's about what? Around 1,000 micromoles, right? 1,000 micromoles, 200 watt per square meter is 1,000 micromoles, okay? So about 1,000 micromoles is saturation is happening. And this is tomato data. But if you look at the canopy, the dense canopy like just we saw, it never saturates. Why? Did you come up with any reason why it doesn't saturate? TJ?
Because of the height difference?
Height difference, yeah, so.
The leaves are getting less light.
Oh.
The upper leaves that are oversaturated, so if you take the average.
Yeah. Bingo. Yeah. Okay, so think about the light intensity 1,000 micromoles, right? Think about the top leaf. They are already saturating. So you come to the greenhouse and then maybe do whatever magic, put the lights or whatever, and then increase the light intensity from 1,000 to 1,500, 50% more increase, right? Top leaf is already saturated, so it doesn't show any increase. But lower leaves, probably 100th of that level or 1/50th of that level, So it's still somewhere like here, lower leaves receiving. So still they have a much more capacity to increase the photosynthesis. So you have so many layers of leaves. So additional 50% increase would definitely increase all the remaining leaves photosynthesis except the very top layer. Okay, make sense? So increasing more light does increase canopy photosynthesis. Any question? Okay, so now you might think about, so the single leaf, it saturates. Canopy doesn't saturate. Is there anything between, right? Because the canopies are many layers of leaves. Different canopies may be different number of layers of leaf, right? So the next one, which is not in your Monday's handout, but it's in a D2L. I uploaded and basically updated the presentation up there. But this one is showing canopy photosynthetic rate. Okay, so the per square meter growing area based photosynthesis. And then it is showing the difference between low density canopy, so less layers of leaves, and high density canopy like tomato canopy. Okay, canopy again is a group of plants growing together, right? So if you see that, the low density canopy is actually saturating. per square meter base, the magnitude is very small compared to high density because it's only, so many leaves in that canopy. But as the canopy is getting denser and denser, more layers of leaves, then you see that the curve is getting more like linear response, right? And then this value, LAI 0.3 all the way to 6.0, is the index we use to express the density of the canopy, how many leaves overlapping. We define this clearly when we learn growth analysis, so you don't need to too much worrying about right now. But smaller the number is, basically less, low density. Leaves are not overlapping. Yes?
What does it stand for?
Leaf area index. Yeah, LAI, leaf area index. Yeah, so see that. So tomato growing around 4.0 or maybe higher leaf area index. So that is a purple symbol in here. So that means, yeah, it's almost linear. By the way, this one is a simulated results. So mathematically simulated based on a given density and given leaf response curve. So we can do that, engineers can do that, this kind of simulation. All right. Okay. So that the canopy response is almost linear, right, to the increasing light intensity. And that is very important because now you don't have to worry about saturation when you consider the total energy coming from the sun in the greenhouse. And then that is the base of the concept of daily light integral. So the total light, regardless of the instantaneous light intensity, whether it is low, it is high, as long as canopy is dense like that and linear response to that light intensity of the normal, range we can get in a greenhouse, all the way from 0 darkness to maybe close to 2000, it's linear response. And in that case, you can actually, get the cumulative value, regardless of the instantaneous light intensity, and express the total amount of light, photon flux, instead of per second, but per day. And that is directly determining potential growth of the day. Okay? It's very important. So I think this concept came from floriculture group originally, but greenhouse controlled environment scientists. So we started using the concept of daily light integral. So basically the unit is moles of photons per square meter base, still per square meter base, but instead of per second, it's per day. So per day, meaning from sunrise to sunset. Okay? Because dark period, it's usually 0. So you can increase the 24 hours, but during the dark time, it's 0, so technically it doesn't count, right? So that's more per square meter per day. And then growth is supposed to be linear when you have a dense canopy to the DLI. More DLI, then you have more growth. And that is the base. If you are familiar with tomato growing, took some courses in CEA, you probably have had that 1% reduction of light is 1% reduction of yield. Or opposite way, 1% increase of the light is 1% increase in your yield. And because it's a linear response canopy has to the light. Okay? Otherwise, you wouldn't say that, right? If it is saturating, you wouldn't say that. So DLI is, again, it's a cumulative value. So it's almost like a rain gauge. And this is exactly explained one of the reading articles you have in a Monday lecture, under the Monday lecture D2L module. And so it's basically want to measure all the photons, right, which have the wavelengths of 400 to 700, because it's a photosynthetically active radiation. You don't want to count anything outside of this range because it doesn't drive photosynthesis. So you are having rain gauge per square meter base and then measuring per day how much photons per square meter per day. And so here is the one, exactly the same one you saw on Monday. Outside level in the green, and one greenhouse is blue, and then the other one is in red, and showing the actual diurnal changes over 2 days recorded in November. And I computed DLI based on this instantaneous. So the instantaneous value is ppf per second value. So every second, if you know every second, then you just sum up, right? This second and next second, you just record it per second and then sum up. Sometimes your recorder doesn't do that many scanning. So in that case, you have to assume if the measurement interval is 5 minutes, then you have to assume this measurement, the first measurement, and next measurement is 5 minutes later, and then you have to assume the value in between is close to maybe the first measurement. So you need to approximate, every 5 minutes. So considering that is the average of the 5 minutes. So 5 minutes, how many seconds? 5 minutes times 60 seconds, right? 300 seconds. So whatever the value times 300 seconds is a 5-minute cumulative photon flux. And then you do that for whole day, you can do that Excel, right? Whole day value and just sum up. And then that would give the daily light integral value. So I got 32 moles per square meter per day from these values about. Outside, inside 17 moles per square meter per day. And then another greenhouse, 10 moles per square meter per day. And then somebody in a class on Monday asked about potential light intensity that required for growing different crop species. Great question. Yes, there is a recommendation. For example, lettuce, you want to shoot 12 to 15. That's a recommendation. Moles per square meter per day. 12 to 15. You could grow lettuce at higher light intensity, but lettuce has some issue under high light, therefore only 12 to 15. Okay, so greenhouse A for lettuce is a little bit too high. Tomato, it's a high light loving, they like high light. So as long as temperature is maintained, they can grow very high light. But researchers are suggesting optimum is around 30 moles per square meter per day, 30. So greenhouse A even is too low, right, for growing great yield of tomato, achieving great yield of tomato. Outside is good, but maybe temperature is not good because it's November, you have cold night, and tomato doesn't like that cold temperature. So see, that's actually the base of DLI recommendation. And then why DLI? Because canopy photosynthesis is very linear to the light intensity. So this one is so University of Arizona has a very nice data of weather data, and then this is a Tucson data, and then it's converted to micromoles. Dr. Giacometti and his staff member did this conversion from the recorded value in the weather station. Weather station doesn't use micromoles. That's really bummer, but they use watt per square meter. So you need to change the unit by knowing the relative or approximate conversion. Usually 1 micromoles per square meter per second is 0.5 watt per square meter. So it's a 2-to-1 ratio. So if it says say that again. If it is saying 1 watt per square meter in the azimuth, weather station, 1 watt per square meter, that means 2 micromoles per square meter per second. Okay? Yeah, you basically double to get ppf, photosynthetic photon flux. But anyway, so then you get the cumulative value, and then this is plotted over the Julian day. So one is January 1st, 365 is December 31st, right? And then just plot it over the that 1 year period, but 13 years of data. 13 years of data. So you see that in Tucson, outside data, of course, is as great as 70 moles per square meter per day, summertime. And in wintertime, even we can get 30, moles per day, per square meter. Of course, we have cloudy day like Monday, and then in that case, very low light intensity, therefore not even 1 moles per square meter per day. So maybe that's typical in northern climates, right? Northern states, Midwest. So depending on the location. So this is showing the difference between the locations. Right-hand side is the same graph, same chart you saw in the previous slide. Left-hand side is in Ithaca, New York, where Cornell University is located. So they have a nice weather station there. But if you look at the data, this is the same 13 years, or not exactly, maybe different range, but about the same duration of data. And then you see the scatter, so that means in a climate like New York State, you have very, very changeable weather. At Tucson, Southwest, dry climate, we have relative consistency, right? So that's why it's very, very dark. It's all along the potential maximum level. So it's very different. And then that means the capacity for the plant to grow is different. So that means growing plants in Ithaca, New York may be unpredictable. And you might need to add some artificial lighting in a greenhouse to grow during winter. But in Tucson, you may not need that. So this is very useful just by looking at light intensity, capacity of plants to grow. This is Dr. Jensen's data, lettuce data. I just wanted to show plant growth is actually linearly responding to the DLI. This is moles per square meter per day, so the daily light integral. And then it is showing lettuce growth is almost linearly responding all the way to 20. But the plants under the 20, I believe it was not as good as somewhere in here, because of the stress. And I think he was referring to oxygen stress in the root zone. But anyway, so it's a slightly different thing. But anyway, another thing here is tomato yield. So in tomato case, this is a yield in the y-axis, cumulative yield per square meter. So how many kilos of tomato, as a total from the beginning of the crop harvested per square meter. So that's the y-axis. X-axis is the cumulative, but not per day. It's the entire period. So this is showing the linearity of the total energy received by the greenhouse, and so the linearity between the greenhouse total energy received, that duration, and the yield. So this is also another evidence. Yes, plant growth and yield are linearly responding to the cumulative light, right? And then not only the growth, most of the case, yield, which is fruit weight, fresh weight, so that's also responding linearly to the cumulative. And this has to be the photosynthetic radiation. All right, any question so far? Okay. So as I showed you the difference between New York State and Arizona, different locations, geographical locations have different DLI, and I believe that second article I put in the D2L Monday has this, lot of charts showing the difference between different states or locations in different months of the year. Okay, so that January and June, so that this is the range. You see that we have a relatively high light all year round. Okay, so this is really helpful when you need to decide the location to build your greenhouse to do a production capacity. Otherwise, you know, so for example, northern climates, you see that, the DLI in January is, I think in here, Midwest, it's about 10 to 15 January. Okay, so you definitely need artificial lighting to add to grow tomato. And that's what they're exactly doing in the Midwest. Also, if you go to this area, it's only 5 to 10 moles per square meter per day. So maybe you don't even grow lettuce in that climate. In that case, it might make sense to grow indoor, you know, completely indoor. Otherwise, you need to put too much lighting in a big greenhouse. So, you know, that it helps to decide what growing capacity they have. On the other hand, you know, so the Tucson, we have a lot of light, 60, 70 moles per square meter per day during summertime. However, too much light, meaning too much heat also. So as long as you can control temperature, plants can, you know, particularly like a high light crop, like a tomato, they can take that high light, high DLI. But usually temperature is the case, you know, prohibiting the high productivity. And then we will talk about temperature, you know, in probably in 2 weeks or so. Okay? All right, so supplemental lighting, artificial lighting to increase DLI when it is small, you know, lower than the recommended DLI. And then this is the way it looks in northern climates or northern European countries during winter time. Okay, so talking a little bit of unit, Caitlin, she can't be here today. She had some other pre-scheduled commitment, but she was asking about unit of solar radiation and unit of solar radiation, whether it should be micromoles per square meter per second or watt per square meter. And my short and quick answer is, if you are dealing with plant response, use micromoles per square meter per second. If you're dealing with energy in the greenhouse, how much heat is coming in energy, you got to express the value watt per square meter. It's very, because energy unit versus photon unit. Photon is the small, the minimum unit that drives a reaction. So that's why, you know, if it is plant response, you have to use photon flux. Yeah, so the rest is not so much important. Okay, any question? All right, so that's the end of the light intensity lecture, and then I want to move on to light quality lecture from now. All right. Okay, so this series has light intensity and light quality, and then now light quality, right? Colors of light. Okay, so to refresh your memory, light quality, we can see some lights, right? Almost the same. So 380 to 780 is our visible light radiation. And plants use a little bit narrower range of light for photosynthesis, driving photosynthesis. So it's kind of interesting, you know, similarity of the two. And then we talked about, you know, how you know, gradually change the color of the visible light. And photosynthetically active radiation and visible radiation or visible light. All right. Oh, need to go touch in here. Okay. All right. So solar radiation or sunlight. Okay, I talked about sunlight unit, right? Energy unit and micromoles. This is actual spectrum of the sunlight or solar radiation. Okay, so x-axis wavelengths. So the distribution of the photons, where they are falling in terms of wavelengths. Question?
Do you have notes?
Oh yeah, sorry. Yes. Thank you for reminding. Yeah. Okay. Good. Thank you. All right. Yeah, so 300 to 3,000 nanometer is the sort of rough range of solar radiation. Some people use like 280 to 2,500 nanometers or 2,800 nanometers because it's very small. It's depending on the accuracy of the sensor, and then also the atmospheric conditions where they are measuring is at high elevation or low elevations. It's affect the edge and then also some minor. So, but in my lecture, I want to stick with 300 to 3,000, one of the commonly used range of solar radiation. Maybe because it's easy to remember, 300 to 3,000, just 10 times, So, but anyway, so it's just showing the distribution, okay? So if you measure solar radiation outside the atmosphere, this curve is very smooth, okay? But because of the atmosphere, particles and moisture and everything, either reflecting or absorbing certain wavelengths of light, so that's why you see, up and down, up and down, up and down. Okay, so that's solar radiation. So you see that PAR, photosynthetically active radiation, which is coming 400 to 700 range. So the much of the energy is in that range, sunlight. And then a little bit of UV and more what we call infrared radiation. Okay, all right. And then when you show the spectrum of sunlight in a whole range, 300 to 3,000, often the expressed unit is a radiation unit, watt per square meter, because plant response is only here. So if you express everything photon flux based, then it's just a little bit weird, because much of the radiation beyond 700, 800 nanometers are basically heat, if you talk about plants. So that's why you see watt per square meter in this range, right? Okay. So if you remember, if you remember the photosynthetically active radiation and the response of photosynthesis, waveband specific efficiency of photosynthesis, you might remember red light has a higher efficiency, which is a quantum efficiency, by the way, so that only the light limiting situation. Highest efficiency is in the red range, and then the second highest in the blue range, and then the third one is in green range, right? Green light. So this is also measured for single leaf, okay, single leaf, not entire plant or entire canopy. And measuring quantum efficiency of whole plant is very challenging. But, first of all, measuring photosynthesis of entire canopy is challenging anyway. You need to have a nice setup and then, all the bunch of sensors. But it is very important to understand this is a McLeod curve, right? So the many plant species average obtained by researcher called McLeod, McCree, McCree, but single leaf base. So researchers wanted to see, what is the canopy-based response, and is the blue and green relationship is still the same in the canopy. So the Dutch research group did this theoretically. So this is the absorption, and this is a quantum efficiency. So, but both are showing the solid symbols as a leaf response. This is a leaf response, and open symbols as canopy response. And what it's showing is leaf response, clearly green light is not efficient, right? Because of the reflection. The leaf has a little bit more reflection in the green range, therefore it doesn't contribute photosynthesis as much as blue or red light. But if it comes to canopy, reflected light has another opportunity to get absorbed by neighboring leaves or lower leaves. Okay, so when it comes to the canopy level, the difference between the light quality in terms of photosynthesis, in terms of photosynthetic efficiency, is very, very small when it comes to canopy. Leaf level, it's very significant, right? You see clear dip, but canopy level, you don't see much dip. Okay, of course there would be in-between situation, right? So if someone can simulate different density of the plants, layers of leaves, LAI, leaf area index, then you should be able to see the gradual change. And high dense canopy is probably almost flat. Okay, so that's why we use 400, 700 without any discrimination of light colors. Consider every single photon contributes to photosynthesis and quantify the light intensity in that range without really worrying about. Make sense? Okay. If you have more questions, let me know. It's actually it's a very difficult concept. It's not so easy. But I can explain that if you want me to do again. All right. So let's talk about the plant response to different colors. So in terms of photosynthesis, as I showed you, it's not much difference in terms of blue, green, red. Red is relatively higher at the leaf level. Canopy, it's still showing kind of the similar trend, but not so much different. Okay? The light quality also affects the shape of the plants. Stretch, stem length or stem elongation, stem extension, or leaf expansion, or maybe the compactness of the plants as a result. Okay, so this guy, relatively old data, but tested under the natural light environment. This group built multiple chambers outside, and of course temperature is supposed to be controlled because otherwise you don't know what you are testing. And then they put different filters over the box, and it created different light environment. So this is a natural light, and this guy had 3 different crops species: chrysanthemum, tomato, lettuce. And then blue filter transmitting blue, and then another filter transmitting green. So that means internal environment is green-rich environment. And then yellow light, yellow environment, and then red environment, and compare the plant response. And then you can see that plant responds to the light qualities. And what he saw, or this group have seen, is relatively small plants when only blue light was applied, or filtered plant. Filtered light quality was rich in blue. Then plants were very small, and this could be explained probably in a way blue light affects the plant. So blue lights limit the expansion of leaf, and then also stem elongation. So make the plants compact, okay? So when it happens, so basically plants don't have much size of antenna, right, to receive the light intensity or the photon flux. So compared to natural or other treatment having a big leaf, under the blue light, because expansion growth or extension growth is limited, so the leaves are much smaller. So total amount that plant can receive, total amount of light that plant can receive is much smaller, and therefore contributed to the smaller growth. So it's not necessarily, in this case, blue light was very inefficient in terms of photosynthesis. It's more like a photomorphological. So the shape is different, therefore the total leaf area is different, therefore total growth was affected. So when you come to light quality evaluation, maybe now, some of you are working on that now, or some of you may be interested in doing that in the future, you need to pay attention regarding the photosynthetic influence of the light and then also morphological influence of the light. So that under certain light, plant can expand the leaf, and then in that case, even though photosynthetic efficiency is the same, because of the larger leaf situation, you may be able to enhance the growth. Okay? All right. Okay, good. I hope we can have more time to discuss this because this is very important. Yes?
Why is green, why is green more efficient? I thought that,
This one is not the efficiency. This one is just showing the shape of the plants. And green, yellow, red seems like expanding or stretching, the stem more than natural light. And then there would be potential factors there, whether it is a UV light or far red, which is helping. So it's not exactly saying what is contributing in this case.
so if you were to go back 2 slides, I think, it was the single leaf response to
Green light.
various light. And I think the highest efficiency was in the red range, followed by blue and then green.
Yeah, so-
Is this the difference between canopy?
Yeah, so the canopy difference is showing in the green range. So if you look at the photosynthetic rate per leaf, per single leaf or per square meter leaf, green light of the leaf is definitely lower. But if you look at the whole plant, whole canopy, then the decrease is not as great as single leaf. Okay, TJ?
So although the green light is less efficient or less effective, it's still.
In terms of photosynthesis, yes. But the particular study I showed right here, it's probably something else is making plants larger, therefore it looks large. And then maybe the fresh rate is larger than natural light. But it may not be green light. So it's green light environment, but it contains more different lights. So maybe far red light and UV light is the ratio. So it's not. So I'm gonna go through how you quantify light quality because it's not just one light doing something. Okay, light quality evaluation is very tricky. Okay, again, it affects photosynthesis. Also, it affects morphology. So that's why some people misunderstand, which light is more efficient. And then, for example, this experiment was done for single plant base. So that means plants are freely, getting the light. But if it is more dense canopy, then the influence of different light quality is going to be different. Okay, because plants cannot expand the leaf size so much because neighboring plants are there. So that would limit the influence. So it's very light quality effect. You got to test in the actual commercial. If it is application in commercial scale, then you have to test in a commercial scale. So otherwise, the data coming from very simple experiment is misleading. But anyway, I hope I have a more time to discuss this. Maybe I'm going to do this in an online discussion, which is actually coming up this Friday evening, and then also Saturday morning with my online students. So if you are interested, you can participate that. Okay, so this is a kind of summary of the different light quality influence reported in a bunch of different textbooks or literatures, journal papers. So blue light is known to affect elongation and then also morphogenesis, elongation, and then the shape of the plant. And then blue light is known to affect stomata opening. When blue light exists, then stomata opening tends to increase. So more opening of stomata if blue light comes in. And blue light and UV light affects the pigment synthesis. So the anthocyanin purple pigments in strawberry, purple or red lettuce, those pigments, or eggplants, those pigments affected by or enhanced by UV light. Red light is reportedly reduce the expansion rate, so make the plants more compact. And violet light is opposite way, enhancing expansion. And then red and violet and blue are known to be involved in phytochrome response, if you remember plant physiology, if you have taken that. And then blue light is different receptor, and UV light is different receptor. Receptor is basically sensing the lights and giving the signal to the plants, do something, stretch or synthesize something. Okay, and then more complicated thing, which is a top-notch science in plant science. Those receptors seems to interact with each other. So that is super complicated plant physiology. So right now, researchers working on how those receptors interact and then what is the result of the plant response. So a lot of study going on. And then now we have a lot more capacity, I would say, to test different light qualities, because if you know LEDs, light emitting diodes, you can create any spectrum, if you want. And then you can test a bunch of different hypotheses. So it is a great era, by the way, to understand light quality response of the plants. Okay, all right. So this is a little demonstration of my team. I just wanted to show that plant pigment develops, different way, or different rate, I would say, production rate of the pigments under different light qualities. So those three plants are the same variety, same species, same age, lettuce. And then the left-hand side, probably a little bit darker might help. The left-hand side is grown under high UV transmitted greenhouse. Second one is a medium UV transmission. We put acrylic panel. The last one is a very low UV, which is a polycarbonate panel. And then we actually put the shade cross too, so that the light intensity, DLI and light intensity are all the same, but only the difference is light quality. So you see that high UV transmitted light enhance the pigmentation, anthocyanin, purple pigments, because anthocyanin synthesis is enhanced by UV light. So it is clearly showing, right, no UV, then you don't have purple pigment. So light quality affects the secondary metabolism of the plants. All right. Okay, so there are different names for different colors or different wave bands of light. So I'm just putting this as a summary. Photosynthetically active radiation, you probably remember now. 400 to 700 nanometer range is the PAR, photosynthetically active radiation. And ultraviolet radiation, there are A and B and C UV light. UVC doesn't exist because it's absorbed completely by the atmosphere. So it doesn't exist natural environment, okay? So UVA, which is close to the blue, so the longer wavelengths, and UVB is shorter wavelengths. So the very beginning of sunlight is covering UVB, right? That is very important light for pigmentation. And then also, you get suntan, and that's UVB effect. And then also very powerful light. So if you irradiate something with UVB, you might, kill the, bugs and germs and those things. That's UVB. It's very powerful because energy level is inversely correlated, right? Not proportionally, inversely correlated with the wavelength. So the shorter the wavelengths, energy level per photon is higher. So that's why UVB is powerful. UVA is more like a blue light to the plants. And infrared radiation is greater than 800 nanometers, particularly when you're working on plants. You want to separate out 700, 800 and name that different way, because 700 to 800 is a far red light, which is very important light for the plants, far red light. And beyond that is infrared light. Courtney, you have a question?
Yeah, I was wondering, the
with the high UV, medium, and very low UV. Which UV did you use? Like the air?
Yeah, good question. Yeah, so the last slide I showed you this one, right? So the high UV is, we quantified a simple sensor, but it is supposed to quantify both UVA and UVB. And for anthocyanin, both would affect. Both UV lights, A or B. A and B affects the anthocyanin development. Okay, and red and infrared is very important to understand phytochrome response. Phytochrome is a photoreceptor of the plants. It's involved in many different response. And typical response is stretch, and then also germination. And then I show you quickly the example of the response that is governed by phytochrome, which is a sensor for the plants to sense the light quality. And then we express the light quality using the red and infrared ratio, which is a very simple way to quantify the phytochrome related light quality, red and infrared ratio. So these are the seedling, young seedlings of brassica, broccoli, growing under different red and infrared ratio. Red light is 600 to 700. Infrared light is 700 to 800. By the way, infrared light it's very hard to see in your eyes because it's very edge of the visible light, right? Visible light goes to 780. And so it's very end, and you can't really see, even though the same photons coming in, it looks so dark. But anyway, so that if the ratio is low, that means more infrared relative to red. Ratio is low, that means more infrared relative to red. Then plants stretch. Okay, and then if it is high, more red relative to infrared, then it gets compact. Okay, it's very dramatic response plants have to red and infrared ratio. This is another old literature, but it's a textbook, picture showing how plants respond to red and infrared. High red relative to infrared make the plants shorter. So the internode length, the length between the two adjacent leaves is getting shorter and shorter and shorter. So as a result, very small, compact plants. Infrared is high and the red infrared ratio is low, right? If the infrared is great, large value, then it reduces the ratio, and in that case, plants stretch, because stretching the internode length, right? So that's exactly how you see in the understory of the canopy. Plants do not absorb much infrared light by leaves. So in the canopy, if you measure the light quality within the canopy, all the lights basically transmitted or reflected by the other leaves. And then that light quality is very rich in infrared. So the shade growing plants tend to stretch very quickly, and because one reason is rich in infrared, the environment is. So I'm going to show you the difference of the light quality, light intensity in the natural environment. So this is an incident light, sunlight, and then obviously it's a midday measurement. The range of the photon flux in this case, this particular table reported by those people, used 400 to 800 nanometer. This is because they wanted to show the total photon flux affecting plant morphology. Okay, not just photosynthesis. So it's not photosynthetic active radiation range, right? It's a little bit longer. Active radiation for photosynthesis is 400 to 700, so it's a little bit wider range, just because they wanted to quantify how much energy in that range. So anyway, so don't too much worrying about, but the range is a little bit wider than photosynthetic range. So 700 direct sunlight, and then if you look at the ratio, so this group look at the blue range, 400 to 500, green range, 500 to 600, red, 600 to 700, infrared, 700 to 800. And then so that almost equal ratio, photon flux, number of photons, 1 to 1 to 1 to 1, almost. So the red far red ratio is of course about 1.0, right? So that's a natural light. If you go into the canopy, it's very different look, all right? Of course, you have to expect huge reduction of light intensity, which we demonstrated earlier today. That if you look at the ratio, huge percentage now in the far red, because plants do not absorb that light quality for photosynthesis. Plants absorb the photosynthetic active radiation, so 400 to 700 is well absorbed, but outside they don't, because chlorophyll doesn't. So that's why it just transmit or reflected. So that's why you see a huge far red. So the ratio is very small. Another interesting point is, so blue is only 4% here, out of 60 micromoles, 4% is in blue, and red is 11%. Incident light was almost 25% across, so it's a huge reduction in blue and red, but not so much in green. Not so much in green. It's still 15%, so higher percentage compared to blue or red. And this is because green light is not, absorbed at higher rate by, by the leaves. Therefore, you see more green light sitting in the canopy. But again, the number one component is far red, right? So now I have soil, 5 millimeter below the surface of the soil, and then also very far red rich environment. Okay, light intensity is very, very tiny though, but still far red rich environment. In the water, its situation is different. Far red is, whatever the reason is, far red light is absorbed very well by the water, and therefore what you see is a very large red far red ratio in the water. Okay, so understand that is very important. This is just a graphical presentation of the previous table so that you see the distribution is very different and the different measurement locations. This one is germination of seed. So it's a classic, the response of phytochrome of plants, red and far red, reversible response. So the red light is promoting germination of lettuce seeds. In long time ago, a group of people tested that. And then if you apply red light followed by far red light, and then that would reduce the germination rate. So the limit, restrict the germination rate. And then if you add red light again, then it enhances the germination. So it's a reversible response. Okay, and then that is actually helped to develop the model of phytochromes. If you remember plant physiology or plant biology 101. So phytochrome is the pigment that absorbs certain wavelengths of light. And there are two forms exist in plant. One is the far red absorbing form. The other one is red absorbing form. And far red absorbing form is the most functional, biologically active form. This is going to be in the nucleus and then start causing some reaction. For example, enhancing germination, triggering germination, or making plants shorter. Okay, making plants shorter. That's another example of phytochrome mediated reaction. And if far red absorbing form absorb far red, they change the conformation, the shape of the molecule to the red absorbing form. Okay, and then it basically becomes not so influential. All right, and then also same reaction happens in the darkness. And then when this guy absorbs red light, or sitting in the light quality having a lot of red light, then it goes to, again, very influential far red absorbing form. So this is quite interesting mechanism exists in the plant, and then you can learn this more by taking plant physiology course if you haven't taken that, or reading the chapter of plant physiology, which is describing phytochrome. Very important function to change the direction of growth, whether it is stretching or it's making compact or germinations. Many different, it's listed in here. More than this involved, or phytochrome is involved in those many plant responses. Okay, so it is very important to understand phytochrome. Okay, let's see. Yeah, so one of the classic response of phytochrome, as I said and as I showed a few examples, stretching the plants. When phytate is rich in the environment, then plants stretch. So they elongate. And because, so if you go back, oops, the other way. So phytate-rich environment make the phytochrome in this form, red absorbing form. So the influence of phytochrome is minimum. And in terms of stretch or extension growth, phytochrome would react as suppressing the stem length. So if PFR, phytate absorbing form exists plenty in a plant system, then plants are not extending much. It's very compact. So that's the action of phytochrome. It's related to the plant growth substance, gibberellic acid, which you might have heard. You know, it's basically promoting extension. But anyway, so the one of the classic photobiology is the phytate influence in the stretch, and also let's see. Yeah, so also we know that end of the daylight is affecting stretch of the plants. Because if you look at the plants usually extend during the nighttime. During the day, because of the light and containing a lot of red light, therefore plants don't stretch. So if you look at the nighttime, you see plants stretching. And that trigger is basically coming from the end of day light quality. And then so for example, this one, I have two different plants. One is grown under red-rich light environment. This is actually artificial light environment, but it doesn't have any phytate in it. So that's a control plant. This is a squash. Cucurbit respond really well to the light quality, so we often use cucumber or squash for light quality response study. But this is showing how influence or how sensitive the plants are to the end of day phytate. So during the day, because of the light exists, plants do not stretch. So it's kind of waiting to to have the dark period. And then as soon as dark period starts, plants sort of wait a little bit. It's a lag time. Maybe 1 hour or 2 hours waiting. And during that time, phytochrome is converting into the red absorbing form. So the limitation for stretching, now it's going to disappear. So as soon as it disappears, then plants start stretching. So that's what happens during the nighttime. But when you apply phytate at the beginning of the night, they quickly convert the phytochrome into the red absorbing form, which is not so active in terms of suppressing stretch. So plants start growing really fast when you do that. So end of day phytate to your right hand side, which is very small amount of light we applied. In this case, 4 micromoles, only 4 micromoles, and 30 minutes. And then after 3 days, this one is stretching like that, and this one is still compact. And then this technology we tested because we were working on vegetable grafting. And vegetable grafting, you really want a certain length to work on. Otherwise, it's very difficult to work on the plants. So because of the efficiency of the work, growers want to stretch the plants. And then after stretching the plants, they want to make the plants more compact. So there are two objectives, and then it's very difficult to manipulate the stretch by growth regulators, like gibberellic acid, because you really can't stop the reaction if you use chemical growth regulator. It can stretch, but you can't stop the reaction at a certain time. So light quality is a great tool because you initiate when you want to initiate, when you stop, when you can stop it. So again, this is based on the phytochrome response. And then at the end of the day, the light quality is a signal for the plants to stretch. Far red ending light quality is actually helping to stretch. And then red ending light quality, or red light environment at the end of the day, make the plants shorter. So that's quite interesting example. And I'm going to stop in here today. It's running out of time.